Actually, I think this topic is very interesting and also very important for many people. I think all of you come to this summer school because you are working on proteomics or interested in proteomics. But I also believe some of you have experience about NGS data analysis. So how to combine these two together will be a very interesting topic. So in order to put everybody in the same page, if you have no idea about NGS technology, I will briefly introduce to you. NGS technology can be classified to two different classes. The first one is DNA sequence. So basically, you lysate the cells, and then you will get the genomic DNA. So this is a very long DNA, and then you fragment it to small pieces. Normally, it's 200 nucleotides or 150 nucleotides or 300 nucleotides. And then this small pieces fragmented DNA, we can call it DNA read. And then we attach the adapter to the both end of this fragmented DNA. And then this adapter can be attached to the flow cell. Then we do the amplified to PCR. Then the DNA can be amplified a lot. And then basically, based on different fluorescence attached to the nucleotide, then the machine can tell us what is the DNA reads are, what is the order of an ACTG like this. So we will get the whole information of this sequence. And then the other thing classes is RNA sequence. So basically, RNA, we have a poly-A tail, so we can use poly-T to pull down this all RNA, genomic RNA, and we also fragment it to a very small pieces, then attach a primer, and then we do reverse transcript, and we can get one strand of cDNA. And do another side, then we can get a complete cDNA. So we also attach maybe barcodes or adapter, then we can also follow the same procedures, DNA sequencing, then we can get the signal or the whole information from the RNA part. And then there are also many different protocols using DNA sequencing or RNA sequencing to achieve different purpose. So I just briefly introduced two of them. One is DNA-based, the other one is RNA-based. This one called ChIP-seq, we can imagine if there's a cells here, and we will have a lot of protein and the DNA, they may have interactions. So we cross-linking the positions which interact with protein and the DNA, and then we digest all the DNAs. So you can imagine if there is a protein bind to the DNA, and then this DNA will be protected. So every DNA which is not bind by the protein will be digested. And then we use an antibody to pull down all this binded protein, DNA-binded protein. And then we also digest the protein. Afterward, only DNA which actually attached to protein will be left. And then we do sequencing. So after mapping, you can imagine the region which interact with protein will highly enriched. So we can understand which part of DNA is interact with our target protein. Okay, the other protocol is ribosome profiling. We will also use this data for our tutorial tomorrow. Basically, we have RNA here, and then the ribosome will bind to RNA in order to do the translations. So we also digest this RNA, which is not binded by the ribosome. And then we also use Perseus to digest the ribosome. And then only the RNA which attach to the ribosome will left. And we do the sequencing, then we can know which region is undertake the translation process. Okay. So this just briefly introduce how useful of RNA and DNA sequencing. So actually use NGS technology, we can do a lot of analysis for different process or different events, different biological phenomena. You can do something for the chromatin, you can do something for whole genome, you can do analysis for RNA part, you also can do something to understand translation part. So there's a lot of things we can do, a lot of protocols that we can use. But however, we can say each omics data, they have their advantage. Of course, they also have shortage. For example, if you want to understand the phenotype changed, it's not able to attack this or detect this in DNA or RNA level. If we want to know the non-coded RNA, which is a very popular issue currently, then you have to go to RNA. If you want to understand SNP calling, you want to know the mutation, you want to do the analysis for methylations, then DNA is the options. So how to integrate all these omics data together? It will be a very, very important issue. If we can really merge this omics data, we believe there are more interesting that we can explore. So in Wikipedia, they do some statistic. Actually, until 2018, you can see the citations of the paper in PubMed related to multi-omics data analysis was increased strongly. Of course, it's not so many. You can see only 500. That means this field is in the initiate stage. So it's a lot of things we still can do and then to explore it. But you can imagine if it continues to increase, there will be a very, very hot topic that we can do. So I believe multi-omics data analysis will be the future. Okay, and there is a bunch of papers related to multi-omics analysis published in numerous high-impact journals. And then these studies, they all use multi-omics, maybe metabolomics with like genomics or transcriptomics with proteomics. There's a lot of combinations, and then they figure out many different interesting things. But because the time I cannot introduce a lot of them, so I will choose one paper to be an example. This paper is from my previous lab. Actually, they use transcriptomics and then proteomics data to discover the protein-RNA interactions or protein-protein interaction complex. Okay, this method called GRASIC. The workflow is very easy. They lysis the cells, and then they centrifuge these two. So you can imagine the heavy molecular weight molecule will be in the bottom, and the light molecular weight molecule will be in the top. And then you also imagine if there are some interaction or complex, the member of the complex will in the same fraction as well. So they take 20 fractions on the tube, and then every fractions they go for single RNA sequencing or go for proteomics. So based on the analysis, we can get ingredient distribution in RNA level and protein level. And then basically we can compare both of them to get a correlation coefficients, then we can really know which protein or RNA they are interact. So there are some example. You can see this the ingredient distribution in RNA level. And then this is ingredient distributions in protein level. They use housekeeping genes as a benchmark. And you can see this 16S RNA is highly associated with 30S ribosome. And then this is another example, like 23S RNA is associated with 50S ribosome. And then if we go to the gel, you can see the 6S RNA also highly correlated with RNA polymerase. And then this is another evidence. You can see in RNA sequencing, these two fractions is highly expressed. This 6S RNA is highly expressed in these two fractions. And then its associated RNA polymerase also is highly expressed in these sections. So there are more evidence from the protein group level here. You can see the tRNA modification factors, like contaminator factors. As long as they are form the same complex, the distribution is highly correlated. Okay, so we can discover protein RNA complex and also protein-protein complex. In this paper, they also discover a new RNA, non-coding RNA binding chaperone, which can bind to hundreds non-coding RNA. Okay, so now like what I said before, there are the experimental designs for the DNA sequencing and RNA sequencing. So how can we do the following data analysis? So basically, we will get raw file from a machine, and then we do alignment to the genome. And then we will get Ben file, which is like the result of alignment, which compressed. And then we also can use this result to generate gene expression table. So actually, the process is very similar as proteomics. We got the raw files, and then we use the raw file match back to the FASTA file, and then we got the intensity, right? So actually, it's quite similar. So there's a lot of database you can download for the alignments. You can get the notation files. You can get the reference genome files. And all these database can use to achieve the alignment. And then in our groups, we have Perseus. And the Perseus can help you to generate gene quantifications tables. And this table can be used to following data analysis. Okay, I will show you all the process how to do the analysis of multi-omics data analysis in tomorrow the tutorial. I just briefly introduce some concept here. Basically, if you go to the Perseus and then there's a icon for NGS data loading, and then you press it, this window will come. And then you can add the files. Basically, we add BAM files this after alignment. And the BAM file can be a lot, and then we'll put here. And then you can decide you want to do strand specific or not. And then you can decide the cutoff for the mapping quality. And then also you need to assign the genome annotation file you can download from any kind of database. And then you can choose which feature that you want to do the quantification. For example, you want to quantify the protein coding regions, or you want to quantify the exons or anything else. And then you can specify how many threads you want to use for the computation. So what is the strand specificity? There's three different options. The first one is non-stranded. That means as long as there's a RNA reads map on the genome, on the genes, then we count every single reads as a number to these genes. Okay, so if you can imagine there are 10 reads mapped on the genes, then the number will be 10, no matter it's sense or antisense. The other option is sense. So we only counted the reads which have the same directions as genes. So if we have 10 reads mapped on the genes, and then 8 of them is the same direction as the genes, then the number of the quantification will be 8. And then the other one is antisense. So in the same case, then the number of the quantification will be 2. So why this importance? Some study, they are trying to understand the antisense RNA or the antisense small RNA, or they want to understand the antisense transcriptional starting site. So maybe this option will be useful for this kind of study. Okay, and then we also can specify the different number of threads for the quantifications. So you can imagine if you have you do something for the human genome, and then you generate like 10 or 20 RNA sequencing data, and then every single RNA sequencing data may have 10 gigabases. Then if you want to do the quantifications, the running time will be huge. So if possible, you can assign the many parallels to run this at the same time, and then the running time can be significantly decreased. Of course, you also need to take care about your memory. But basically, if you increase more threads, the running time will be decreased. Okay, now I want to introduce you some tools which we use for quantifications. Basically, there's a paper called HT-seq. This tool is widely used. People use this to do gene quantifications. And then basically, they just do what I said before, like how many reads actually map on the genes, and then give them a number of it. And then we also use the same idea, and then put into the Perseus. And you can see the correlation coefficient between Perseus and then HT-seq count is almost 1. So that means the result is almost the same, okay? But if we check the running time in Perseus, can significantly decrease the running time. So basically, we can help you to reduce your time to generate the quantifications. Okay, so now we have quantification tables, so we can do a lot of things. We can compare different omics data. For example, in this case, we compare the transcriptomics data, translatomics data, and the proteomics data, and then you can calculate the correlation coefficients. Basically, the translatomics and transcriptomics data correlation coefficient is higher, as you can see the 70%. Basically, because you can imagine that ribosome profiling, actually they are RNA-based stuff, so they should be high. And then like proteomics and then transcriptomics, the correlation coefficient is not so high, like 36%. This is quite common in genome-wise, because there are a lot of events where a factor will influence or cause the difference between these two omics data, like post-transcriptional modification, like different half-time of RNA and protein. And sometimes the RNA have some mutation or knockout, and then maybe need to take time to influence the protein level. So this cause this not so highly correlated. But still, we can do a lot of things based on these comparisons. Of course, we also can do only two omics data. So for example, here in translation and proteins, we have the experimental set, and also we have a control. And you can see in translation process, the genes green part is not changed a lot. But in proteins, they are huge, downregulated. So what happens here is also very interesting that we can discover. Of course, we also can do simply to classify the data to see how similar of our samples. Just easy to use like PCR or something. So this is already built in the Perseus. Okay. Now we already show you the data that we can easy to compare. But actually the most common things for the data analysis is differential expression analysis, no matter which omics data you used. If you have experience for using Perseus to do differential expression analysis, you will notice there's not many options that you can choose. Basically, we integrate the student t-test. You can compare one sample, two samples, or multiple samples. But nowadays, there are a lot of tools which can do the differential expression analysis for RNA sequencing data. And it is mature and it is really, really useful. So one example is DSEQ2, okay? Actually, of course, you know this too, so they should have a DSEQ1, okay? So in DSEQ1, they already cite like almost 9,000 times. And then DSEQ2, they cite like 10,000 times. So you can see this only published in 2014. Until now, only 5 years, they already like explode. So this tool is very useful. They can detect the differential expression analysis very precise and also provide a lot of information. This is another tool called HR, which is R packages, and then you can install it in bioconductor. Also, many people use it. The citation time also have 11,000 times. And then the above two tools actually is focused on RNA sequencing, but actually RNA sequencing data is the continuous data set type, but in proteomics is not this case. So there's another tool called Lemma, which is originally built for microarray data. So actually it's quite similar as the data type is quite similar as proteomics. But right now they also developed some new algorithm which can use this Lemma to RNA sequencing as well. So this one is the algorithm, and then you can see the citation is also very high. So basically in Perseus, we integrate these three tools into Perseus. Sorry, I have to say this, this plugin you cannot find in your current versions. Currently, we are trying to working on a paper which is a tutorial paper for plugin in Perseus. And then when the paper out, all these newly developed plugin will be released as well. So at that moment, you can really use it. But actually, original this kind of package is from R or Python. You also can use that in Python or in R. But we build it in Perseus, will be convenient for you to use it. So basically, there's a plugin called DSEQ. DE analysis means differential expression analysis. And then if you press it, you will see a run DSEQ analysis, and then you can choose these three tools for you. And then each tool, they have different parameters you can play around. You can assign different normalization way or something else. And then you can assign like which group you want to compare. If you have control, you can put in the reference. If you have experiment, another condition, you can put in target, and they can compare with each other. And then you can set the cutoff for fold change, log2 fold change. For example, you set 2, that means the log2 fold change is higher than 2, then we say this gene or this protein is up-regulated. It's like you say -2, is the other way is down-regulated. You can set the cutoff of FDR or Q values, like 0.05, to see, okay, which is gene is differentially associated. If you only compare two samples, maybe you also can set p-value as a cutoff. Okay. And there are some limitations of this differential expression analysis tool. For example, in DSEQ2, they're based on binomial distributions. That means they only accept integer. This is not a problem for RNA sequencing because RNA sequencing, the quantification is based on the read count. It always should be integer, but for proteomics, if you do some logarithms, they might become not integer anymore. So the good thing is maybe we can do some adjust of the value. For example, we round the value become an integer, then we can use DSIC2 as well. And you can also do something more like subtract, or you can divide, you can add constant. But just be careful, you should understand what you are doing because you are manipulating the data, okay? So basically, I just suggest to round the data only. But if you know what you are doing, you can also play around this data. Okay, and then we also can do something to normalize the different omics data. For example, in proteomics data and for the NGS data, the range is totally different. For example, in NGS data, sometimes we got hundreds reads mapped on the genes or 1,000 reads mapped on genes. But in mass spectrum, you can see like 10,000 or even 100,000 of intensity. So this range is quite different. So if we didn't do any normalization, just directly compare these two different omics data, then you will find out all these proteins are upregulated because they are really, really huge. So this plugin can help you to normalize the mass spectrum's range to the NGS range. Then you can easily to compare. Of course, you can use this score, then everything will be standard. Okay, and then we also can remove some genes which only have very few reads mapped on it. Normally, people set 10 or 20 reads. If not more than that, we will discard because this actually is not expressed. Okay, after all this setting and you run DC analysis, then you can easily to use scatter plot to plot volcano plots. For it, you just need to set the x-axis. Just need to set the x-axis as a log2 fold change, and the y-axis is the q-value, minus log p-value or q-value. Then you can plot this, and then you can change the color to label the differential expressed genes or proteins. You can play around this and generate a beautiful picture by yourself. Okay, so I think until now, I already introduced all these plugins. I just want to show you a paper in the end. I think previous months ago, we received a mail from the author. And then he told us that really appreciate our work. They use MaxQuant and Perseus. They watch a lot of videos and the tutorial, and then they're trying to do data analysis by themselves. And then eventually, they published the paper to do the multi-omics in Nature. And then the most amazing thing is none of them from our groups. So that means they use the cell by themselves. So that means Perseus and the MaxQuant can be used by the user who have no bioinformatics background. So that means you want to do the multi-omics data, you just need to understand how to use Perseus and the MaxQuant, and you can do it by yourself, and you can publish on Nature. But it's amazing, right? So we are not really help them, but they really do a great job there. Okay, so I think I'm finished. Thank you for your attention, and happy to take questions.